Where is the off button?
This is off.
It's my great pleasure to continue the lectures under sectional committee presentations. First of all, let me welcome you all after the long break. And now we have the second speaker, Dr. Subhadeep Chatterjee from CDFD, Hyderabad. I have known his work since quite a long time. He has been working on the crosstalk between bacteria and plants, and then the title, as he has shown today, is quite exciting. And he has a very distinguished career. He won Santi Shwarup Bhatnagar Prize in 2020 in Biological Sciences, and he was elected Fellow of INSA and the National Academy of Sciences. And he is also he has been awarded National Bioscience Award for Career Development, DBT, then member Goa Research Conference, Innovative Young Biotechnologist Award again from DBT. With his excellent academic background, as I said, he has been working on the implications of fundamental work on bacterial disease management, which sounds quite interesting because he's going to also talk perhaps on using these findings for medical applications, and also how it can be used to understand microbial evolution and so on. So Dr. Subhadeep.
First of all, I would like to thank for giving me the opportunity to talk in this. Yeah. So I will. So as I will tell you, I will not go much into the molecular aspect. I will show you the secret life of microbes. Like humans, microbes have developed a language system. So as I title of my talk is, we will try to understand the social communication or language between the microbes, particularly those which causes infection and those which are there in the environment. So this talk I have divided into 4 parts. So in the first part, I will show you different forms of communication in the animal kingdom, and how microbes, which are unicellular organism, has developed a communication system to tell each other and to communicate with each other. And that will be the first part. In the second part of my talk, I will tell you about their secret language. What is their language? Whether they speak Hindi or Bengali, or what is their mother tongue. And the third part, why they talk with each other. So we talk with each other for understanding and communication, and also to get lot of news. So what they are talking about, why they talk. And in the third part, I will tell you 2 aspects of talking, that if they talk, there are certain advantages to them, and if they talk, there are certain disadvantages to them. So how they manage both the advantages and disadvantages. And in the fourth part, last part of my talk, I will tell you some applied work which we are doing. Basically, we are a basic biology lab. So by understanding the basic biology of the microbes, can we devise some strategies to stop disease? So if you see the communication, so response by communication is very important for all living organisms. So different aspects of being called as being alive, like growth, reproduction, response to change in environment is a very crucial factor. So organisms must respond to the change in the environmental condition. For example, if there is too cold, the organism try to avoid the cold, and that happens due to communication with each other. So if you see evolution of language, so language, many people, linguistic biology, linguistic people compare it with a evolution of humans. As human evolved, language also has evolved. For example, if you see the example of English language, people have quoted that like human beings, there is a evolution, both selection as well as variation, which happens as the language travels, as the human population, civilization travel. So, if you see different mode of communication by different organisms, for example, firefly blinks. Nowadays, you don't see fireflies in the cities, but if you go in remote area, they blink, and they blink in a particular pattern to give a signal to their for the mating. And for example, the honeybee dance. So, actually the honeybee, when it is collecting the honey, by the dancing language it tells the other fellow mate where the sweet flowers are. So, they tell the direction. So, there are different mode of communication, some by smell, some by pheromones or chemical molecules. But when it comes to bacteria, this is a picture of a single bacteria. You see, it looks like a very lonely, alone living organism with a size of 1 to 4 micrometer. So this is what the bacteria they have called as prokaryotes. So they thought of it, they have a very lonely life. And a field of biology was developing. So they started asking the question, do these tiny organisms which are 1 to 4 micrometer in length, can they also communicate? Do they also have a social structure like humans or higher animals? So the clue came from a very indirect experiment. So actually some marine biologists who, like NIO, they are hosting us. So those are the marine biologists situated in Hawaii. So they were studying this squid called a Hawaiian squid. So this squid lives in a Pacific Ocean, and this squid emits a light through its light organ, and that was quite fascinated by the biologists where the light is coming from. Later on they found out the light which is produced by the squid is not its own light, it is produced by bacteria which colonizes the light organ of the squid. So this bacteria colonizes the light organ and produce light. And the second very important point, they found out that they produce light only when they are in large numbers. That means when bacteria is less in number, they don't produce light, and when they are in large in number, they produce light. So they got interested and tried to see what is the nature of this light communication happening. So as they grow the bacteria in a flask, when they grow, as assumed that at a low cell density, at a low number of bacteria, they didn't emit light. At medium cell density, when the bacteria grow more in numbers, they start producing light. And at very high density, when the population became very high, they started talking with each other. That means they all started producing light. So they got very interested what happened when they were high in number, why they were not talking when they were less in number. So they did a very clever experiment. So what they did, they collected the supernatant which was collected in the flask without the bacteria of a high talking bacteria, and then they put in a non-talking bacteria. Then everybody started to talk. So that indicated there are some message or chemicals which are produced by the bacteria when they are high in number, and if you give the message to a low in number bacteria, they can also detect the message and talk. And then the whole field of socio-microbiology started trying to understand the social microbiology or social life of a bacteria. So now people started seeing the bacteria not only lives in a very solitary lifestyle, they undergo transition of a very complex lifestyle. So this is a confocal, for example, I am showing a micrograph where these mountain-like structures are actually biofilms which are formed by millions of bacteria which come together and form a mega colony. And this is kind of a housing society. And this is switching. So from a solitary to a community lifestyle and again to the solitary lifestyle. people got very fascinated how bacteria decides when to live alone or when to build a home. Then the whole concept of this quorum sensing started from this marine bacteria. So this is what the whole concept of quorum sensing. It is a density dependent cell signaling. As the bacteria grows in numbers, they started producing chemical molecules which are called as language molecules. Okay. So this production of language molecule is a density dependent. Higher the density, more the molecule will be made. At a particular threshold, when the density reaches a particular threshold, it tells the bacteria, now you are this much, now you do this thing. And in what to do different things, regulation of gene expression in a synchronized way. So this is what is called as synchronized gene expression, action in unison, that means together. And that actually by this bacteria can count each other, how many we are. And actually in the lab you can see them speaking. We cannot hear because they make chemical molecules. So what we use in our lab, different biosensors which can detect different language molecules. So here you can see plate full of bacteria, they are talking. Here the reporter gene has been hooked to a language responsive promoter, and here you see they cannot talk. And they actually love to talk with each other when they are together. And what are these different languages? So as discovery started about the bacterial language molecule, it appeared it is much more complex than even human. So for an example, this is most studied language molecule called acyl homoserine lactone. So I will give you a very basic example. They have a lactone ring and a acyl change. It is very similar to our own language like Sanskrit. Sanskrit is the base and different dialects are added. Okay. So now suppose this is the base language. Now this bacteria speaks like Bengali. Now this adds a other chain, it speaks in a different language. So in this way there are many peptides and also borofuranol. I will not go into in touch, but the main family of signaling molecule which our lab works on, that is called as again a fatty acid like signaling molecule, very similar to the language, which is called a DSF, diffusible signal factor. So my whole of my talk will be based on these molecules. And now, is the language speaking is so simple? One bacteria can speak one language? No. Actually, this is an example of Vibrio cholera, which causes cholera in humans. So it speaks in two different languages. Why? Because this language which is for talking with themselves, and this language is for talking with other bacteria which is there. So one is their private language, one is their common language. So the private language they do private things, and the common language they communicate with the other bacteria what they are doing. So this is very complex. And some bacteria are much more clever. They don't speak or they don't communicate with any language, but they have ears or receptors where they can hear the language from the other bacteria which is producing. Okay, very similar to our noisy neighbor cartoon, in which when some new neighbor comes, the other neighbors are more interested who has come in. So they want to know their neighborhood to cause disease. And this is a very simplistic model of bacterial quorum sensing system. I am giving you in a very simplistic way. So basically there is a synthase which is there, which makes the quorum sensing signaling molecule, which is a diffusible signal molecule, and there is a DNA binding receptor which is there. And binding of this receptor to the DNA depends upon the concentration threshold of the signal. More bacteria is there, more amount of signal will be there, then the receptor will be bound and then it will regulate the expression. In that, this is a very basic mechanism, but it is far more complex. I will give you a hint because of the lack of time. Since it will be a general talk, I am giving you in a simplistic way. And now the main question comes, why they talk? What is the necessity to talk? Imagine a situation, suppose there is one bacteria firing a bullet to break the wall. Suppose imagine the wall as a host barrier. The firing power of the one bacteria is so less that it will not able to break the wall. But if many thousands of bacteria fire together, then there is a great chance that the wall will break. So this is the whole concept of quorum sensing. So there are certain tasks which if performed together gives much more advantage as a group rather than the individual. So individually you may do the task, but the benefit will be not that much. So therefore the whole concept of quorum sensing as a group behavior, social behavior is important. And so in our lab we work on a group of pathogens called as Xanthomonas. This is very important group of plant pathogens which causes very diverse kind of diseases in different economically important crop like rice, cabbage, and even citrus and many other crops. So what we found out many years ago that there is a signaling molecule which the Xanthomonas makes, which we call it as diffusible signal factor. The most interesting part is that if you don't have this signaling factor, it loses its ability to cause disease. So this is the disease symptom which is there in the rice, and this is the rice leaf infected with a wild type bacteria which can talk, and if you take a non-talking bacteria, it is not able to cause disease. So our whole research then started why this talking is so important to cause a disease. Then we started seeing the bacteria inside the plant, and what we found out that these bacteria, these are in green labeled with a green protein, and this red color are the xylem vessels. Okay? So those are the vessels of the plant which conducts water from the root to the other parts. So this bacteria actually colonizes the xylem vessel, and when we looked in the xylem vessel, you see they form a very macro colonies or biofilm like structure. And if you imagine a lifestyle of a pathogen, if a pathogen has to infect a plant, you can imagine first it has to land on the leaf surface. This is a cartoon showing the leaf surface. Then it has to find its suitable spot, and then it has to enter inside the tubes or the vessel. If you compare the size of this bacteria and the leaf, it is uncomparable. It is in micrometer, this is in centimeter. So it will take its lifelong journey, even it will die to find the spot. So what actually it should do is that it should find its way when it is in low cell density, and it should colonize inside the vessel when it is in high cell density. So there is a two switch. There is a low density switch, it tells the bacteria to attack, go inside. When they go inside and becomes enough in number, then they tell them to form a biofilm. So as we have identified, there are many biofilm forming factors which I am not going into much detail. So these are absolutely essential for the bacteria to colonize inside the plant. And quorum sensing, if you take a mutant, it is unable to form a biofilm. And this is just a movie. If I have time, I will show you. So this is actually the wild type bacteria which is moving, and this quorum sensing system actually stops the bacterial motility. It regulates the flagella to form a biofilm. Now you see the mutant, if you see, they are moving super fast. So there is no control. So the message is not there. So they are moving around and they are not forming a biofilm. And you can add the signal externally and again give back to like the wild type. So as I was telling you, this pathway is very complex. So through lots of genetic screens and mutant analysis, this is kind of a overall regulatory circuit. I will not go much into a detail, but as you can see, this is not a very single parallel phenomena. There are multiple transcription factors and response regulators and two component systems which are involved in Xanthomonas, which we identified many. This model is built by us through our understanding of the basic mechanism of understanding how pathogen actually communicates with each other. And this is the overall biological model. So at low cell density, when less quorum is there, it gives a signal to the bacteria, now you move and activate your chemotaxis system. And once it get inside, it starts the second switch, which we found out, that it activates cell wall hydrolyzing enzymes, which tells the bacteria now you extract nutrients. I told you one picture that if you are together, you fire bullets, the damage will be more and nutrients will be released, and the number when it increases, it tells the bacteria now you form a protective biofilm or a big colony, so that you will be protected against the harmful defense of the plant. So these all are fine. So far I told you bacteria has a communication system, they communicate with different type of language, and why they communicate, because there is a gain advantage in doing things together, and there is complex signal transduction system. But one evolutionary question which arises is that, so quorum sensing, as I told you, it's a density dependent benefit, because if you are more in number, there are always benefit to talk, but for every system there are disadvantage. So it is a double-edged sword. Imagine a situation if these are a group of bacteria which are talking with each other and performing tasks. Some cheaters will arise in the population. So these are mutants, okay. So they don't do quorum sensing, but they take all the benefit from the other people. So soon these people will be also turned to cheaters, because they think they will get more benefit. The cheater has not studied in the exam, but he is passing the exam. So other people will also cheat, and soon the class will collapse. But it never happens in the system. So how the system controls the cheaters, that I will tell you. And the second thing is, too much of talking is also bad. Two thieves are talking too loudly and they can alert the host system. So too much of language talking is also bad. So now I will, third part of my talk, I will talk about how bacteria manipulates and counters these two burning edge problem. So what the theory has been, that the evolutionary theory predicts that there is a division of labor is always good. So if you have a division of labor, you have phenotypic heterogeneity to perform bet hedging survival strategy. So I will give you a very simple example. So at the end, so what they are telling is that if all the cells are doing the same job at the same time, if suppose situation now changes, now everybody will be not tuned to change to that situation. So therefore the population keeps heterogeneity among itself so that they can switch to the change in the other lifestyle. I will give you an example. Suppose when corona happened, now one person, there are 4-member family. Now one is bread earner and 3 are dependent. And one is bread earner, he is working on a computer from home, and the three are the dependent people, they help in the family. Some help in cooking, some help in the cleaning. But the 4th person doesn't do anything. He enjoys and takes all the benefit. Now it's a corona time. Suddenly his computer fails, the bread earner's computer fails. Now that the 4th person who doesn't contribute to any in the community, he is very good in this computer. Now for his own benefit, he comes and fixes the bread earner's computer. So this is called as division of labor. So the population maintains heterogeneity. There are performers and non-performers. So the non-performer don't perform at the time, but they do certain functions which are not done by performer, and that is beneficial at a particular environmental condition. I will show you the first time in bacteria what we discovered is called as reversible non-genetic heterogeneity. So as I was telling you that quorum sensing synchronizes population to perform tasks in unison, but as I told you that evolutionary theory predicts that phenotypic heterogeneity in performing social tasks is beneficiary, as I gave the example of a bread earner and that thing. Whether it happens in bacteria, so we started asking this question. I am not going much into the experimental detail. Basically what we developed is synthetic circuits to detect the quorum sensing system in the bacteria and to ask the question what individual cells are communicating. We are seeing as a whole population now how the individual cells populate are communicating. What we observe that there is a significant proportion of population in the group which are non-responding to the quorum. So those are those cheaters which are there along with the performers, and these cheaters do a function which is called as helps in the bacteria to move away from the biofilm. And if you take either the cheater or the performer, again you analyze them. So this is separating the cheaters and by molecular facts, and then you can analyze, and what we found that again a cheater can give rise to performer and cheater. So that ratio in the environment is maintained 80-20, and we discovered that that switch is an environmental switch which is the nutrient switch xylose degrading enzyme, which is the xylose, it limits the bacteria to utilize xylose, but since performer produces the xylose, so when xylose is the only nutrient source, then performer outcompetes the non-performer. So this is one movie I wanted to show you. So this is the life cell dynamics we do along with the, so you see now these two are performing, and this is, now you see they have already performed, now it is generating performers and non-performers. So these are hyper performer and the non-performer. And what we found out, which is not yet published, is that we discovered, we have found out that this bacteria also has a short term memory. So up to 6th generation they remember that we are not performing, but after 6th generation the memory erases, and then again it gives rise to 80-20 ratio of performer and not performer. So this is what the model which we gave is that during quorum sensing it tells the bacteria to perform tasks together, but to maintain heterogeneity it maintains certain proportion 20% of non-responding to the quorum of cells. In a biofilm, when there is a high quorum, these performers promote biofilm formation and the non-performer escape from the biofilm because they don't respond to the quorum. This happens, we showed in planta also, that there is an advantage in having both population for the systemic spread of the pathogen. So if the pathogen remains in a biofilm, it will be stuck at that stage, and if it maintains certain non-performer, they will again colonize to a new niche and they will again form a performer non-performer. So bacteria maintains two different populations, that gives advantage of the bacteria to escape, host defense, as well as switching between talking and non-talking population. So now this is happening all in the bacterial front. What happens in the host? Host is also clever, so they are also developing certain strategies to stop. So what we found out in the course of our work, that the host have developed a immune system to detect the bacterial signaling molecules, and it can mount a defense response. So at the initial stage when the bacteria is colonizing, it primes the plants to produce the innate immune response, and when the innate immune response is generated, bacteria again is more clever. It secrete effectors or the bacterial molecules which can suppress the plant defense response by chelation of calcium. I am not going much into detail. So we have shown the molecular mechanism, like, so this development of the communication system I showed you, they are communicating, they are maintaining some certain cheater population to perform multiple tasks. Host has developed mechanism to recognize, and bacteria has again developed mechanism to suppress it. So this happens in a model in which now when the bacteria attacks the host, this is the plant cell and the bacterial cell, so it makes the DSF signaling molecules, and this DSF gets detected by the plant, and plant induce MAP kinase based defense response, and the bacteria cleverly suppresses it by secreting extracellular polysaccharide which specifically chelates calcium, which is important for induction of defense response. So these are fine, many interesting aspect of the understanding the life of bacteria. Now, whether we can do some applied work in understanding this biology. So, a article came in Scientific American. So, they talk about that antibiotics are prone to generate resistance. This is very simple. Suppose antibiotics is there and there is one target, and if there is a mutation in the antibiotic target, then the bacteria will become resistant. So, you see now more and more dose of antibiotic is required, and bacteria are coming multi-drug resistant. This is because due to accumulation of selective mutations which is gaining resistance. So, then a group, very big group in UK and US, now they are trying what is called a social medicine. Now, you develop certain inhibitors or something which can stop its social life instead of targeting the bacteria. So, I will skip this thing because of lack of time. So, this is an example how you can attack the social life. So, bacteria takes up iron, now you put a dummy or a analog, and in that way it will form a locked complex and it will be not taken inside by the bacteria, and bacteria will be starved of iron. So, what we are talking about, whether we can interfere with the quorum sensing system to develop strategies to reduce disease. So, one strategy is if you can degrade the quorum sensing signaling molecule, gone, bacteria are not able to communicate. The second strategy is another strategy which we call it as a loudspeaker strategy. For example, if you are giving a talk here and you are talking, now somebody plays a DJ music in front of you. Now, nobody will be able to understand and nobody will want to talk. So, the thing is that if somehow you can make the signaling molecule in large amount in the host itself, then the bacteria will be fooled that we are too much in number, we will stop the infection. And these are what is called as social drugs, as an antimicrobial agent, because it is not targeting one system, it is targeting the whole pathway of quorum sensing system. So, we adapted two approaches, one is the transgenic one and one is the non-transgenic one. So, transgenic one is that we wanted to synthesize the bacterial signaling, quorum sensing signaling molecule inside the plant. So, in that what we did was we make a synthetic circuit with all the components of the bacterial signaling molecules and put it in the plant, in the chloroplast, localized it to the chloroplast. Because why we choose chloroplast? Because chloroplast is more similar to bacteria if you compare it evolutionary. And another was the non-transgenic one that I will show you. So, what we did, as I told you, we developed the circuit of the quorum sensing system, all the synthase or the enzymes which are required, and codon optimized it to work in the plant, and we transformed the plants. And now these plants we also tested biochemically, they have started producing bacterial signaling molecules. Now we wanted to see actually what they respond in the infection. So, in the y axis, if you see, this is the disease index, is the number, is the index is the indication of the severity of the disease. So, this is the disease caused by the wild type plants. When you inoculate the bacteria, heavy disease, all the transgenic line show very high rate protection against the disease, and when we check the bacteria in these plants, they were restricted towards the point of entry, because this plant was making so much of DSF molecule, they gave the signal to the bacteria that you are too high in number, you form a biofilm at that stage. So, unfortunately in India, the transgenic work is not that encouraged in the government, because now they are starting CRISPR-Cas system, but that is also for manipulating endogenes. So, but this now has been adopted in California. Now they are trying the same thing in grapes. So, that is also happened from our work. So, but we are now not doing in the field, because it is not allowed in country to work with the transgenic plants. So, what we did was a non-transgenic approach. Now again we develop the whole synthetic circuit and put it in a beneficial bacteria, and in the lab you can produce a large amount of these signaling molecules, and you can spray it with a surfactant. So surfactant are the chemicals which actually penetrates inside the leaf, and when you mix it with the synthetically made or a biotechnologically made the signal, and you can spray it and this gets incorporated, and you see this also gives a very large protection. So this is an example of a leaf which has been sprayed with a buffer, and then pathogen has been inoculated, and this is the disease symptoms, and this is the leaf which has been sprayed with the artificially produced DSF signal, and now this gives a very large protection. So now we are collaborating with Directorate of Rice Research, and we are talking with them to make it as a formulation. If in the agriculture it may work or not. This is the result of a greenhouse. In the field many things can happen. So we are still not know, but at least we demonstrated that interference of the social communication system can hamper the disease process. And in US also now in Pseudomonas aeruginosa infection, people are using this social medicine as a target to prevent lungs infection in cystic fibrosis compromised patients. So this is the last movie I will show you. This is talking, cheating. So these bacteria are talking with each other. This is the signal output from the communication. Now they will go into a cheating mode. I will show you that how these are the always performers. So they are performing up to 6th generation, and this is the cheaters which came up, which are not performing. So they are always talking with each other. They are more clever than us. So whatever we make, they break after a certain time. So that's the interesting part of microbial world, and I would like to thank the past members in our lab who have worked in lot of projects. So we do in our lab many projects. One is the host pathogen interaction, like the DSF signaling in the plant. What are the plant components which are used to detect bacterial signal, and the whole performer and non-performer synchronization work was done by Binod and Bishwajit, and the other people work on different aspect of virulence mechanism of using Xanthomonas as a model system. And we also use quorum sensing as a model system to understand population altruism and cheating using a prisoner's dilemma. So you make certain blocks of different mutants and then test that how fit they are. If both are cheating, they are more fit. If one is cheating and one is performing, the system is more fit. Thank you.
Thank you, Subhadeep. I can see already quite a few hands. Professor Nagaraja to begin.
As usual, very clear and very lucid presentation. But the question is regarding your cheaters.
Yeah.
From this, this perhaps in dictyostelium it is perhaps very clearly laid out, but when it comes to bacterial cheating versus non-cheating, I don't know whether there's a clear-cut definition. Perhaps it's a bit of a gray. Perhaps in Myxococcus xanthus there is more evidence. And in your last video, it is more, there you can't clearly distinguish.
No, actually this is just an example.
Okay. That didn't look like a real distinction.
No, actually what we do microchannel entrapment and then study because of the lack of.
Then, in the cheater population, what you see in Myxococcus, I'm sure you are xanthus or there is a very clear phenotypes are
actually
very clear phenotypes, and also there is a genetic signatures.
I will show you.
Yeah, Okay, if it is there, it's fine.
Yeah,
but
Actually, because of the, see, it's a very interesting question.
it is not real black and white.
Yeah, it's not a real black and white. I will tell you two aspects of the very interesting question Dr. Nagaraja asked. So there are two types of cheaters. One is called as the genetic cheaters, and this is called as phenotypic cheaters. So genetic cheaters, what happens in Pseudomonas aeruginosa, where it infects the lungs. So people have found in hospital samples, when they take out the bacteria from the lungs, infected lungs samples, they found many of a spontaneous mutants which come up in the quorum sensing system, but they always remain in a selective pressure of using the private group 80-20. So they are genetic cheaters, fine. But the reversible non-genetic teacher is a stochastic switching between 80-20. The ratio remains the same. A bright cell and a black cell comes in the ratio of 4:1. Now if you take a black cell, which I showed you in the segregation analysis, this will again give rise to black cell up to 6th generation, and then it gives rise to again black and bright in a 4:1 ratio. So this heterogeneity factor is more like a epistatic type, not a phase variation type. And the switch, what I didn't showed you in that in our recent work, is that there is a nutritional switch. So if you have nutrient rich media, the 80-20 ratio is maintained, and there is as soon as you remove the nutrient rich condition and put them in the plant mimicking media and put xylose as a whole carbon source, now we found out the quorum sensing actually regulates xylose utilizing enzymes, and that xylose utilizing enzymes are intracellular. So even performer and non-performer is there. If the performer is there, it is making the food eating apparatus inside itself. So therefore it will get benefit when xylose is there, but the non-cheater will not. So at that condition they are wiped out.
Good, Subhadeep. I think one question was very much relevant to what Dr. Nagaraja had asked. Now, when you say the cheater, when it divides, it actually produces both population.
Yeah.
Xanthomonas has got plasmids.
No, this one doesn't have plasmid.
Xanthomonas, it doesn't have plasmid.
It doesn't have.
Okay, so that's one possibility I was looking, that if there is a asymmetric cell division,
So we
and you get a population which has got the secondary genome elements,
sir,
that's contributing.
Yeah, very interesting question, sir. Actually, we probed this thing. So it is not asymmetric division. What we found out is, actually in the model, I didn't show you in the model. So it is a stochastic prion-like thing. So there is a cyclic di-GMP is there, which is an intracellular secondary messenger. Its distribution in the daughter cell is very different. So that actually generates the stochasticity.
Okay, all right. I think I have quick other things also. You said a transgenic you made it. The DSF is a single gene, one genic pathway?
No, it is, sir.
From bacteria you put into the plant.
Yeah, so sir, there are 3 enzymes are there. What is called coordinatase hydratase and enol-CoA hydratase. So these are in a operon in our bacteria. So what we do, we stitched in took all those things in a plant promoter and codon optimize it. So now the whole operon is operational in the plant, but when we did a nuclear transformation, it didn't work. And when we then we thought of chloroplast, and because we found out that these enzymes actually is very similar fatty acid composition like in chloroplast or in mitochondria. So chloroplast we thought, and then we put a chloroplast localization signal in front of all those things, and then we showed that in the transgenic plant now it started producing.
Subhadeep, can quorum sensing bacteria be considered as multicellular organisms?
It's the biggest question. Actually, the socio-microbiology people are telling they are the biggest eukaryotic like. Now people have discovered nerve-like transduction happening in a biofilm, Like a recent, sometime back, a Nature paper came in. They thought that biofilms actually are making tubules, a canal-like thing, and through that potassium dependent that signaling is happening. Electrical senses they are synchronizing, and even the morphogenesis, like Myxococcus xanthus, sir has pointed out, is an excellent model system to study the unicellularity and transition.
Just to interrupt, bacteriocellularity is a way of life in bacteria.
Way of life.
Often thought it is unicellular, but actually
It's a way of life actually.
lifestyle statement.
Sir, if you open your like shower, so there is biofilm. Medical devices, you open biofilm, not a single bacteria is there. The most of the cathedral infection which happens in hospitals is due to biofilm. In the
Biofilm is a multicellular.
Can I
yeah, yeah.
So you said that when the cheaters they started accumulating little mutations, right?
Yeah.
So in case of antibiotic resistance, are all of them become cheaters?
No, madam, what happens when in a antibiotic resistance, what happens? Bacteria has a multiplication cycle very fast, 20 minutes or 30 minutes, or in case our bacteria 1 hour. So what happens? It generates variants in the population. Let's say you are talking in the population of 10 to the power 9 cells. In that, if therefore there is a one spontaneous mutant which has arisen in that receptor, now when you put the antibiotic, 10 to the power 8 cell will be wiped out. Only that cell will survive, and that will be selected in the next round. So they will overcome the system. But in this case, what happens is that if you target the quorum sensing system, suppose bacteria has generated a mutation in the, let's say, receptor. Now what happens, next time it will not sense the quorum sensing system. But not sensing the quorum sensing system is disadvantage to its own survival of its pathway, because lots of other iron metabolism, other things are linked. So this is the difference between a antimicrobial and a social drug.
Yeah, maybe the last one.
Subhadeep, how much similarity is there between quorum sensing bacteria that infect plants and the ones that infect animals?
Sir, very similar. Actually, the family of signaling molecules which I talked about, which we work in our lab, is called a DSF. So DSF family of signaling molecule, here again the language is different. Ours is, let's say, Telugu, since I am in Hyderabad. So now there is a Pseudomonas aeruginosa and Burkholderia and Stenotonomonas. They are all opportunistic pathogen in hospital infection. So they also communicate with a variant of DSF, which is called as BDSF. B is Burkholderia DSF, and SDSF, which is the Stenotonomonas DSF. So there are other bacteria respond to this, like very similar to this family of signaling molecule. The other family of signaling molecule which I talked about, acyl homoserine lactone. Even many plant pathogens have acyl homoserine lactone, and Pseudomonas aeruginosa also has an acyl homoserine lactone, and even in other gram-negative bacteria.
Yeah, let's have
You see, I'm sorry,
No, sir.
I didn't catch the ultimate function of the signaling system.
The ultimate
question,
function.
for example, this single cellular to multicellular.
Yeah, I will show you, sir.
Okay, quickly now, if you
Very
look, for
quickly.
example, at amoeba,
Very quickly.
say in certain environmental conditions, thousands of these amoeba merge into each other.
Yeah.
You know, some part is catching food, some part is distributing food, but there is a small portion which is fruiting body which reproduces.
Yeah,
but
yeah.
there is no disadvantage because all of them are 100% genetically identical.
Yeah,
They are produced by one. So if the signaling system here is, the function is to produce more,
See, this is a
then it should be more intense at low population densities rather than at high population density.
Yeah.
So what is the ultimate function?
ultimate function is, one is, I told you. So your question is that what is the ultimate function at high cell density? That's okay. So for that, okay, I showed you a cartoon in which people have done experiments also.
The breaking the wall is fine. You can just tell it.
Yeah. So actually what happens is, sir, certain, let's say suppose the bacteria has entered. Now if they are in less in number when they enter inside the host, if they start showing their weapons in the beginning, then it will be wiped out by the host. Imagine a bank robbery happening. If suppose one or two thieves come, they can be easily empowered by the bank security. But if they come in a group, then they can overcome the security and get the benefit. So the same concept as quorum sensing, like quorum sensing tells the bacteria what to do at what time. That is the most important. When you are less in number, hide your weapons. When you are medium in number, take out small weapons, not alert the host, get the nutrient. When you are high amount, now damage the host. So this timing, spatial and temporal regulation is actually controlled by this quorum sensing. This is the advantage. And the second question of your is the thing what they do, cheating and non-cheating is. So if you see this, I didn't show it you in due to lack of time, but if you have cheaters and non-cheaters together, yeah. So now you can develop permanent switch off and switch on cells by genetic manipulation. Now you see what happens if you, this is a cheater, this is a non-cheater. So cheater promotes biofilm formation. So this is a strong biofilm, and this is the plate actually is showing motility. So the cheaters are more motile as I was showing you, and the performers tend to form the biofilm. When you mix these two and then follow their trajectory, like you see as you go away from the center, this is more the biofilm and this is more migration. You see more and more of this white is the non-performer are towards the systematic spread, and these performers promote the colony formation. It is just like a house in which ration is limited. So suppose 4 members are there, they will prepare a biryani. Okay. So now 10 members have come. Now they tell the 4 members, now you go and find some other house. That's
Yeah, okay. I think a fascinating lecture. So thanks, Subhadeep.
Thank you.
And let me also thank INSA for arranging this kind of lectures, and thank the host. And I should also acknowledge and thank Subir, who had first lecture. And thanks for your all patience. Thank you.
Thank you once again, sir. And today's session's ending here. And we are going to have high tea at 5 p.m. at YBS Moti Auditorium, and followed by the culturals by 5:30, and which will be followed by the dinner at 8 p.m. Thank you. Thank you all. Vehicle is there outside.